So in this next video, we're going to be talking about cell signaling. So we've talked a little bit about this when we were talking about how single cell bacteria or single cell amoeba could respond to temperature changes in their environment to change the amount of unsaturated fatty acids in their plasma membrane. But we're going to talk more today about how cells in multicellular organisms communicate and how they communicate when they're very distant from one another, so when they're at different places in the body. So for an example, let's consider when you hear a loud noise or you're scared. This is sensed by your neurons, and then this sends a signal to begin producing epinephrine. And then epinephrine could be received by different cells in the body to mobilize glucose and give you the energy for a flight or fight response. So distant cells communicate, as I just said, by using signaling molecules. And sometimes signaling molecules are generically called ligands. So sometimes I may be referring to a signaling molecule and use the word ligand. Different groups of signaling molecules include neurotransmitters. These are signaling molecules that regulate the nervous system. And hormones. Okay, hormones are signaling molecules that are sent to distant organs and regulate physiology and behavior. Signaling molecules can also include growth factors. These provide signals for cells to divide, and cytokines, which are signaling molecules that regulate the immune response. And so today we're really going to consider the signaling molecule that are hormones, okay? And so hormones are categorized based on their ability to cross the plasma membrane. And so we can categorize lipid hormones as lipid soluble, like a steroid hormone like testosterone, or insoluble like epinephrine. And so we're going to take a look at these two different signaling pathways. But already you're probably starting to think that those that are lipid soluble might be able to pass through the membrane, and so their receptor is probably inside the cell. And those that are insoluble have to probably rely on some sort of protein or receptor on the outside of the cell to begin sending their signal. So let's first take a look at this lipid soluble signaling molecule pathway. And so here let's consider that this is testosterone, and this is carried through the blood, and it's attached to a protein, right? Because it's not very hydrophilic. But once it gets to the surface of the cell, that hormone can diffuse right through the plasma membrane. So stage 1 is the arrival of the signal, and stage 2 is signal entry. Right? So this goes straight through the membrane, and it binds to a receptor in the cytosol. And when it does that, it causes the receptor to change shape. And so this is a really common theme in signaling, and we call this signal reception. And so you can think of this as here is our quarterback, and they've thrown this signal, right, straight through to a receiver, and then that receiver, right, is going to go right into the end zone. And in this case, our end zone, right, is the nucleus. So this shape change may have exposed a nuclear localization signal. So it might have been hidden in the interior of this protein, and then when it bound to the ligand or bound to the hormone, this caused a shape change. So now important can see the nuclear localization signal, and then this can be brought into the nucleus where it can regulate gene expression. So we have the arrival of the signal and signal entry, and then we have signal reception and signal response. Okay, so very few steps in this signaling pathway when something can pass through the receptor or pass through the plasma membrane. So very short, easy play that ends in a touchdown. Signaling gets a little more complicated when we start to think about these lipid insoluble molecules. Okay. So I want to talk about the steps here. So the first step is that our extracellular signaling molecule binds to a receptor protein in the plasma membrane. Okay, so this is our first signal reception. Okay, so here, right, we're passing this ball, right, to a player, right, maybe a running back. Okay, and I'm sorry for all the football analogies. Okay, this binding causes a shape change in this receptor. And now this is going to trigger a cascade of events that we refer to as signal transduction. Okay, so this is an important term. So we have signal reception, and then we have signal transduction, because what we're doing here is we are changing the language by which this signaling molecule is communicating, right? So we're going to transduce this signal. So it's going to convert the extracellular hormone signal into an intracellular signal. And when it converts this, it will often amplify the signal. Okay, so one player, right, now becomes a little bit of a Ponzi scheme where one person can enlist a few more people, and then those people can enlist a few more people. And so you amplify this signal, okay? And this lets the body get away with producing very low levels of hormones to produce a very strong intracellular response. This pathway, just like the other pathway, eventually leads to a signal response. And often this can be changes in gene expression, but it can also be a signal response that causes movement or the mobilization of stored vesicles. But we're mostly going to think about how signal reception leads to changes in gene expression. So let's take a look at this with a real pathway, okay? And the pathway that we're going to really focus on is this pathway called G protein coupled receptor pathway, okay? So this is a G protein coupled receptor. And when these were originally identified, they were called serpentine receptors, because each one of these in our body has 7 transmembrane domains. And so this protein snakes through the plasma membrane several times, right? Snakes through it 7 times, because you have the seven transmembrane portions, and then you have these intracellular motifs and these extracellular domains, okay? And the ligand is going to bind in the extracellular space, and it's going to transduce the signal into the intracellular space. We also call these G protein coupled receptors, we call them allosteric proteins, because when their ligand binds, they're going to change shape. So let's show that here. I'm showing the ligand as this green circle, and we can think of this as epinephrine binding to the beta-adrenergic G protein coupled receptor. And you can see, right, that this protein has changed shape, okay? And sometimes we depict this as a receptor with a little star to show that it's been activated. Now this is going to transduce a signal, okay? And the signal it transduces, right, is part of its name. So G protein coupled receptor. So it's actually coupled to a G protein. And the G protein that it's coupled to is actually a trimeric G protein. So there's 3 parts of this protein. There's this alpha, beta, and gamma subunit. So that's why it's called trimeric, okay? So just to review, we have our G protein coupled receptor. When the ligand binds, it changes shape, and then it's coupled to a trimeric G protein. Okay, so recall that G proteins are only active when they're bound to GTP, okay? And they're inactive when they're bound to GDP. So initially, before the ligand bound, these were all associated, and the alpha subunit is bound to GDP, okay? But what this receptor is going to do is it's going to promote the exchange of GDP for GTP. And so this alpha subunit is going to bind to GTP. So now the alpha subunit has GTP. And this is going to change the shape of the alpha subunit. and it's going to cause it to dissociate from the beta gamma subunit. Okay, so let's do this one more time. We have our G protein coupled receptor, the ligand binds, it changes shape, right? This shape change helps the alpha subunit of the trimeric G protein exchange GDP for GTP, and this causes the alpha subunit to dissociate from the beta gamma. Okay. Now, next is where I think it kind of gets a little more interesting, because this alpha subunit is going to go ahead and activate an enzyme. And the enzyme it activates, and you don't have to know the name of this enzyme, but it's called adenyl cyclase. And so this alpha subunit activates this enzyme. Now, this is the important part, is that this enzyme is going to lead to the production of a secondary messenger. Okay, so let's just take a second to define what a secondary messenger is. It's a small non-protein molecule that relays and amplifies signals received from the surface receptor. Okay, and the surface receptor is the primary messenger. Okay, so there's several features of it. It's non-protein, and it's usually able to amplify the signal. Okay, and so in this case, with this example, adenyl cyclase is going to convert ATP to cyclic AMP. And so here's what the cyclic AMP looks like, right? So the ATP has lost all of its phosphates, and now, all but one, and then it's connected, right, in this kind of cyclical fashion to the sugar. And this cyclic AMP is a secondary messenger, okay? And this is going to go ahead and activate other proteins, okay? I'm going to give one example of what cyclic AMP activates, but they aren't restricted to one role, and several may act in a single pathway. So I'm only focusing on cyclic AMP, but several could be acting downstream of G protein coupled receptors. And so examples of these small molecules, right, include things like cyclic AMP, as well as calcium ions that are stored in the endoplasmic reticulum. This should say DAG. So DAG and IP3 are lipid signaling molecules. So they're non-protein small molecules that amplify signals. Okay, so these are lipids, this is an ion, and this is a metabolite of ATP. So what does cyclic AMP do? Well, it activates, so it binds to this regulatory subunit on a particular kinase, and I'm not going to give you the name of the kinase, but it then activates that kinase. So what kinases do is they add phosphates to other substrates. So once this is active, this kinase is active, what it does is that it's going to be able now to move into the nucleus. So a nuclear localization signal was exposed. And it's going to go into the nucleus, and it's going to phosphorylate transcription factors. And then those transcription factors, once they're phosphorylated, can turn on different gene expression. And so we have ligand, a change in shape, that facilitates the exchange of GDP for GTP, right? So that the trimeric G proteins break apart. This activates an enzyme that leads to the production of secondary messengers, including cyclic AMP. And cyclic AMP activates a kinase that leads to gene expression. A common theme I'll point out is in signaling pathways, we see a lot of kinase activation. So adding phosphate groups to proteins is a really easy way to turn proteins on and off very quickly. And so it's quite common to see kinases in pathways, and we'll take a look really quickly at another pathway that really only uses kinases. So if we start with a single molecule of epinephrine, right, this can bind to a G protein, right? So we have one GPCR, right, binding to epinephrine, can actually activate hundreds of G proteins. Okay, so there's an amplification event there. But each G protein only activates one adenyl cyclase. Okay, so here we have a lot of G proteins, but each G protein activates only one adenyl cyclase. But then each adenyl cyclase makes many cyclic AMPs. So cyclic AMP its production represents a signal amplification, right? We're turning up the volume on this stereo, right? So that we can really produce a strong response. Just to give you some numbers, the blood levels, just kind of for 1-to-1, of epinephrine are 10 to the negative 10th molar, so minute amounts. And the cyclic AMP levels inside the cell, once that cell has bound to epinephrine, is 10 to the negative 6. So that's 4 times higher, right? So we've really cranked up the volume. And so this helps make this process far more sensitive. So I said we triggered a response, a gene response. And so what would that response look like if we were considering the effects of epinephrine? Okay, so these bind to a group of receptors called adrenergic receptors. And there's different adrenergic receptors on different cells. But in theory, in overall, they're all going to trigger some metabolic changes, okay? So in liver and muscle cells, binding of epinephrine to their adrenergic receptors is going to stimulate glycogen breakdown. So it's going to cause glucose to be released, and it's also going to increase, the body's going to increase blood glucose levels. And the more glucose means the more ATP. In muscle cells, those cells will also start upregulating the enzymes in glycolysis. So those are some of the genes turned on. And so, again, this all helps make more ATP, more energy, so that you can run away. If epinephrine binds to heart cells, this increases the heart rate by vasodilations in the blood vessels. And it also increases, if it binds to cells in the lung, it increases the respiratory rate via bronchodilation. So this means more oxygen, so that this oxygen can be used by the electron transport chain to make more ATP. Now, I don't expect you guys to definitely memorize this list, but I just want to give you a connection to how a signal, right, can transmit into something that's useful for a multicellular organism. So every signaling pathway needs to be regulated. We don't need to break down all of our glycogen, right? Some of it needs to be stored, right? We only need that short burst. And so I just want to talk about some ways that this pathway can be turned off. So every time an alpha subunit turns on an adenyl cyclase, its GTP will be hydrolyzed to GDP. And so what will happen is then when it's hydrolyzed to GDP, it will actually reform as a trimeric G protein. Okay, it can be reactivated if that hormone is still bound, but it can only activate one adenyl cyclase. Okay, so that's short-lived, seconds to minutes. So that's one way the pathway can be regulated, right, as this gets turned off. The other way is that cyclic AMP can also be converted into an inactive product. Okay, so this is AMP, and so you can see that the cyclic portion has gone away. And this happens via an enzyme. Also, right, the hormone or ligand can dissociate from the receptor, and this would change its shape back so that it wouldn't be able to associate with the trimeric G proteins. So these are some ways that this pathway can be naturally turned off. And then the last way I'll talk about is that there's a lot of proteins in the cells that are called phosphatases. And phosphatases have the opposite activity of a kinase. So these are kind of two opposing proteins because this is an enzyme that removes phosphorylation. And so phosphatases can take off the phosphate from the transcription factor that's causing the responses. So this pathway can be naturally turned off. The whole signaling pathway is quite dynamic. So in a cell that's signaling, the numbers of receptors can change, the ability of the receptor to bind to a hormone can change, and the sensitivity to a hormone can change. So this is a very dynamic process that's very well controlled. Signaling can also be actively blocked. And this is why I chose, or one of the reasons we talk about G protein coupled receptors, is because 1/3 of all FDA approved drugs target a G protein coupled receptor. So it's a pretty pharmaceutically important pathway to know about. And so, for example, we can add a competitive inhibitor. So in the case of epinephrine, we can add a beta blocker. So recall that competitive inhibitors bind within the active site of the protein. So they bind where the hormone would normally bind. And if you bound here, this wouldn't transduce this shape change. And so you wouldn't get activation of your trimeric G proteins. You wouldn't get activation of your cyclic AMP. So you wouldn't get, we call this downstream in signaling. You wouldn't get any of these downstream pathways happening. And this drug is used to reduce the strength of heart contractions to lower blood pressure. There's different types of inhibitors for G protein coupled receptors. We can get allosteric inhibitors that bind somewhere else in the protein and shut it off. And we can also inhibit downstream signals. So for example, if we inhibited adenyl cyclase, right, it wouldn't matter if there was hormone around. If we inhibited adenyl cyclase, we wouldn't get any cyclic AMP. So that's what I mean by inhibiting downstream signals. So just to review, hormones that cannot diffuse against across the plasma membrane, they bind to extracellular proteins that transduce the signal into the cell. The signal is amplified and it ends with a signal response. Okay. So I want to talk really briefly about one more type of receptor that uses this kind of pathway. Okay. Oh, a little more here. So the hormone binds to the GPCR. It undergoes a conformational change. That's the signal reception. The signal transduction is the activation of the G alpha, which associates from the beta gamma and leads to cyclic AMP production. And the signal response includes the release of glucose stores, glucose in circulation, and increase in glucose metabolism. Okay, but here's the other pathway I wanted to talk about, okay? So I don't expect you to memorize this pathway. If I were to ever ask about it, I would give you a picture of the pathway and ask questions about it. Okay, so you do have to know the G protein coupled receptor pathway, but you don't have to memorize the steps of this pathway. So this is called an enzyme-linked receptor pathway. And the most popular one is the receptor tyrosine kinase pathway. Okay, so again, what is a kinase? This is an enzyme that adds a phosphate to a substrate from ATP. Okay, and again, a phosphatase is an enzyme that removes phosphates from a substrate. So this is a really important pathway because this pathway is what receives growth signals. Okay. So these here I'm depicting something like epidermal growth factor, okay? So this helps epidermal cells grow. And what happens is when this growth factor is added to cells with these receptors that recognize it, this causes the receptor to come together into this pair. And we call this receptor dimerization because it forms a dimer. When the receptor is dimerized, the signal is transduced because this turns on the kinase. And so it's able to phosphorylate itself. That's why it's called a receptor tyrosine kinase, because it phosphorylates its tyrosines. Okay, so this next step, what happens is once these phosphates are on this receptor, it creates a docking site for bridging proteins. And those bridging proteins activate another G protein, okay? And this G protein is called RAS-GTP, right? So it's going to be active when it's bound to GTP and inactive when it's bound to GDP, Okay, so this is showing that it's active and bound to GTP. And this is going to cause a phosphorylation cascade. Okay, so it's going to activate kinase 1, which is going to phosphorylate kinase 2. That's going to become active and phosphorylate kinase 3, which is active. And this is going to mediate a response. And the response is cells dividing, cells growing. Okay, so this is called a phosphorylation cascade because we have these phosphorylation events that occur in succession from one another, and this leads to growth. And so here's the picture in the textbook. Okay, so I could ask you something about this pathway, like what's a phosphorylation cascade, or what would happen if you took away phosphates from one of these proteins. Okay, but the reason I did want to talk about it is because RAS, which is that G protein in this pathway, is super important in cancer. So more than 30% of all human cancers are driven by mutations in RAS genes. Okay, so it's especially prevalent in pancreatic cancer where there's a mutation in KRAS in 95% of all of those tumors. So very important in pancreatic cancer, and designing therapies that prevent the overactivation of RAS would be really beneficial in cancer treatments. So to review the RTK pathway, the binding causes the binding of a signaling molecule causes the receptor to dimerize. The RTK is phosphorylated, RAS is activated, and that leads to a phosphorylation cascade, and the signal response is cell growth. Okay, so the last point I want to make about signaling is that signaling doesn't happen in real life. Signaling doesn't happen where there's one signaling molecule in the body and one receptor on the cells. So it's crucial to realize that cells receive and process almost a constant stream of different signals, and that these signaling pathways are connected in a network. Okay. So we've just considered signaling molecule binding to a single receptor and then going A1, 2, 3, 4 cell response. But we have signal crosstalk. That's what it's called when these different cells or signaling pathways are connected in a network. So let's compare what would happen, okay, if we have a response for A, right? And we also have signaling molecule B. Let's say these are going, A and B are going at the same time. Well, B would normally go B 1 through 4. But if signaling molecules A is around, you can see that it actually inhibits B. Okay, so this is going to shut down that pathway. So that's A plus B, where you don't get pathway B. But if A and let's say B and C are there, right? Well, you see C actually stimulates a later part of the pathway in this B pathway. Okay, so here we can have A and cell response A. Let's say that's glucose metabolism. And then cell response B is glycogen storage, right? So because you've hit this later part here of the pathway, let's say this is an enzyme that got phosphorylated, you can get both pathways occurring at the same time. So we'll talk more about this in class, but that's it that I have for this video, and I hope everyone has a great weekend. Bye.